Good morning, everybody. It's morning. As you can tell, the sun is shining in my eyes. Ooh, blinding. Today we're going to talk about evidence for evolution. I kind of don't like the name of this set of slides because it's not just evidence for evolution. There's a lot of other material that's covered in this, but we'll go with the name evidence for evolution. All right, so as Darwin comes up with his theory of evolution, we start noticing, or this is part of what made him come up with his theory of evolution, is some of these things that we're going to see here. Some of it is more modern, so some of these things are things that have been noticed or discovered since the time that Darwin came up with his theory. All right, so the first one I want you to write down is homologous structures. Remember, the prefix homo means same, and this is referring to structures that indicate they have a same ancestor. So they're similar features in different species that indicate they have a common ancestor. The most commonly used example are the structures of the arm in many diverse species. So if you look at a human hand, you have the humerus, the radius, and ulna. There's two bones here, and the phalanges are the fingers. If you look at a cat, it has the same bone structure. It's got a humerus, a radius, an ulna, and phalanges. And that's not that surprising. You can kind of see the similarities between, say, a human arm and a cat leg. However, the surprising one is the whale, the penguin, the manatee. They have flippers or wings that are very, streamlined in shape. But if you dissect them, you see that they also have a radius, an ulna, the wrist bones, and phalanges. And the question is, why do they have those bones? I'll give you a contrast to that. Sharks, which are fish and are not closely related, they don't have any of those bones. So if you dissect a shark fin and you dissect a dolphin fin, in a shark fin you just find cartilage. In a dolphin fin you find phalanges, the humerus, the radius, and the ulna. And so it's an indicator that the dolphin is more closely related to humans. Dolphins are mammals, not fish. Same thing with whales. They are mammals. Same thing with bats. Bats are mammals. Birds are not mammals. However, we think that they're more closely related to us than fish are. So indicating a common ancestor. All right, and then, and then this page is comparing gorillas with an ancient, I don't want to call it a human because it's not a human. It's called an Australopithecine. So this is an organism that lived on Earth prior to Homo sapiens, which is what humans are. But they're highlighting here the homologous structures between the two. Just pointing out really quickly, you can tell that the organism on the right is not a Homo sapiens by the shape of the head. The forehead is flatter, so they think that brain development in the front of the brain is part of what makes Homo sapiens a unique species and causes the skull, the mutations together of more brain matter and a skull that protrudes forward is part of what allows us to think in ways that some other organisms cannot. The large jaw is a mutation from our small jaw, so our small jaw is the mutation. We don't need as large a jaw now that we cook food. So there's several things that you can see in that organism that make them different than Homo sapiens. All right, vestigial structures, please write this down. Vestigial structures are features with no apparent purpose in the modern organism, but they may have been useful to an ancestor. And examples include the whale pelvis. That one's weird. We'll get to that in just a second. Leg bones in a snake. What? And the human tailbone. The human tailbone, maybe many of you have heard of it. It's called a coccyx. In Minnesota, lots of us break our tailbones when we slip on the ice. Does that mean that humans used to have tails? Absolutely not. Humans have never had tails. I mean, unless there's some bizarro mutation, but that's not something that was part of human ancestry. The idea is that our ancestors had tails that weren't human beings. And so over time, if we're not needing a tail, we have to eat. When we eat, we feed our body. And if we have a tail, and I'm not saying as a human being, as a monkey or whatever, some other organism, if we have a tail and the tail isn't being used, you still have to eat for it. You still have to feed it. So if someone somewhere along the line has a mutation where there's no tail, then they get to eat less and they're still just as fit because the tail wasn't being used. So mutations that cause structures that are unnecessary to go away, that's an advantage for an organism. So same thing with the whale here. We know that whales are mammals, and we believe that they were once four-legged creatures. Now, does that mean that whales were ever four-legged creatures that walked on land? Absolutely not. That's not what I mean. Their ancestors were wolf-like animals, and that's a great exaggeration, but whatever. They were small, four-legged animals like a wolf, and they lived on the land. And over time, they moved into the water, and mutations that caused the whale ancestors to become more streamlined allowed them to swim better, and they were more effective at feeding underwater, et cetera, et cetera. But one of the things that has remained, that has never totally disappeared from the whale anatomy, is a pelvis. Now, a pelvis are the hip bones, and the point of a pelvis is to hold on to legs. Well, whales don't have even flippers on the back end. Why is there a pelvis back there? And so the indicator is that their ancestors had legs, and mutations caused those legs to go away, just like you've seen mutations in human beings that are missing legs. But it was advantageous for them and considered an adaptation, whereas for a human, missing legs is not an adaptation. Same thing with the snakes. You can see that within their underneath their skin, up against their rib cage, there are small bones that are sort of rudimentary limbs, so small limbs. And those are mutations that cause the limbs to go away, but snakes became successful navigating their environment, and they didn't need the legs. And so then they weren't feeding their legs, and that contributed to their fitness. All right, other things that are evidence for DNA or evidence for evolution that Darwin knows nothing about, similarities in DNA and proteins. Now that's modern genetics. Taking a look at that, he didn't know what DNA was, but it says down below, give me a second to move my picture. This is kind of a quote. I changed it a little bit. When comparing DNA sequences and proteins between closely related species, and in the example, you're looking at humans and primates in this example, the pattern supports the idea of slight modifications or mutations over time. So if you look at the DNA, you can see the Homo sapiens across the top, but then when you look at the chimp, the gorilla, the orangutan, and actually a dog, you see there's more and more and more and more mutations that are causing them to be a little bit different. Interesting fact, we have way more DNA in common with a dog than we have differences in DNA. Remember that dogs have eyes, ears, mouth, nose, heart, lungs, muscles, bones. We have way more in common with a dog than we have that's different than a dog. So that's why we have so much DNA in common with them. All right, number 4, another piece of evidence for evolution is the fossil record itself. So Darwin was familiar with the fossil record, and so these are things that came up as he was talking about evolution. It shows gradual changes in some structures over time, and we refer to those gradual changes as gradualism, brilliant name, and it occurs often over hundreds of thousands to millions of years. It's a very, very slow process, and we refer to those transitional fossils, we refer to those changes as transitional fossils. I didn't say that right. And my picture is covering up, if you're looking at the notes yourself, my picture's covering up, but this is the formation of feet. Oh, somebody's coming in, so give me just a second here. I'm going to pause the video. All right, back again. Sorry, I got interrupted there. All right, so transitional fossils, what you're looking at is the formation of the horse foot. I'm trying to find my arrow. There we go. Still got the sun blaring in my eyes. So this is super interesting. Like you can see, this is the ancestor of the horse, and you can see gradual mutations and changes in the foot. It starts out with having 4 toes, then there's a mutation. Can you see this one toe that's missing? So now it's 3 toes and then a tiny little nub of a toe. 3 toes, 3 toes. Now the 2 side toes become less and less apparent with mutations. And finally, they end up walking on really 1 toe with just a tiny little nubbin of a toe left behind. All right, so that's called gradualism or gradual transitional fossils. However, there were other things also that they noticed in the fossil record, and this is after Darwin, not before Darwin. Sometimes evolution was occurring way too fast. They were thinking that evolution was going to happen on hundreds of millions of years. And occasionally you'll see an organism leveling out, meaning there's no change, there's no change, there's no change. The fossils are the same, the fossils are the same, the fossils are the same. And then suddenly there's this dramatic shift in the fossil, and the morphology, the shape of the body is totally different. And the question is, how is that possible? So that is something that they've noticed, and it's called punctuated equilibrium. So that's a word that you need to know. And they kind of came up with this in like the 1970s, I think, so quite a bit after Darwin. There are many possible explanations for punctuated equilibrium, but I'm going to give you just one example. It may be due to mutations in genes that control other genes. So we just got out of our gene expression unit. You know that there are genes that control other genes. So there are genes, for example, that are called Hox genes. And these are, we never talked about these are master controller genes that determine how the shape of the body is, where the legs form, where all the bone structures form. If you mutate a Hox gene, vast majority of the time you mutate a Hox gene and you're going to have a severe disability. Take a look at this picture here. So this is a fly, and these are the positions where the Hox gene says they're supposed to form. But a mutation in a Hox gene, this is one example, can cause legs to form where the antennae are supposed to. So that's a terrible mutation. That's not at all good for the fly. But it shows a dramatic change where an entire body form is forming in a different location. And is that one possible explanation for why sometimes evolution is quick? If it were an advantageous change, possibly. But we call that punctuated equilibrium when it happens quickly. All right, so other things in the fossil record, bunch of things that I want you to be aware of. So the first thing is the idea of law of superposition, and we've had that since prior to Darwin. The law of superposition says that the oldest fossils are going to be found on the lowest levels. And so if you look at this diagram here, whatever's down on the bottom, I can barely see here, those are the oldest. And we refer to that as relative dating. So we're saying, relatively speaking, the starfish here, where's my arrow? I can't even find my arrow. There we go. The starfish are older than the leaves because they're lower down. All kinds of things affect relative dating, though. There's erosion, there's uplift, some of you know from junior high science, and it screws up the strata. And in addition, how much older is that starfish than the leaves? Is it 100 years older? Is it 100 gazillion years older? And we didn't know. So a while back, we invented something that's called radioisotope dating. And I don't remember, I say a while back, it was at the 1970s, 80s, somewhere in that ballpark. So what they discovered is that radioisotopes decay. That means they fall apart. You don't need to know the specifics. Predictably, they are like clocks, but like the most accurate clocks you can imagine. So they decay predictably with time. So older objects that have radioisotopes in them, the more of the radioisotopes have decayed. So they have smaller amounts of the isotopes than younger objects do. So we refer to the amount of time that it takes half of the sample to decay. We call that the half-life. So I'm just going to give you a grossly simple example. Let's say we have 10 grams of carbon-14. That's a radioisotope. I have 10 grams of carbon-14. After 5,730 years, I will have half of that. Give me a second because somebody's knocking at the door. All right, sorry, I didn't mean to interrupt myself there. People coming and going from my class. Okay, let's move on. Let's try a radioisotope problem. What's interesting, and I will show you, just take a look at the diagram. I am going to grossly oversimplify how to do radioisotope dating. We're just going to use real simple math. It requires logarithms because it is an exponential decrease, which means that it's not a straight line. It's a curved line going down. But I figured out a really easy way to do some math just to make the point. So here we have a story problem. This guy here is called Megaloceros giganteus. I'm trying to get my picture out of the way. There we go. Here's my radioisotope problem. So it's made up, but it just makes a point. Using thorium-230, which has a half-life of 75,000 years, the Megaloceros giganteus was found to have died about 375,000 years ago. True story. What percent of original thorium was found in the remains? So let me show you how to solve a problem like this. You'll never have to do this on an AP exam. I'm just trying to show you how radioisotopes work. So if the Megaloceros had been alive today, it would have 100% of its thorium-230. Now that doesn't really make sense, but just go with it. It would have 100% of it if it had died today. However, it died 375,000 years ago. So the question is, how much is in it now? So if we take a look, if we go backwards in time, sorry, I'm having trouble talking. If we go backwards in time for the half-life, 75,000 years, it would have half of the thorium-230. If we go back another 75,000 years, this is the tricky part, it's half of the half. So half of 50% is 25%. If you go back another 75,000 years, it has half of that, 12.5%. Keep going back 75,000 years, and eventually, when you land at 375,000 years, you know that they found about 3.125% of the thorium that should have been in that fossil was actually in that fossil. Hope that made sense. All right, now we're going to talk about cladograms and phylogenetics. Hopefully you've done the PBS Trees of Life activity, so you're familiar with cladograms. Cladograms and I suddenly forgot what they're called. Anyway, look at structures in the fossil record and that are considered innovations. So we're looking at structures that are new features, new mutations that serve some kind of a purpose. These characteristics appear in later organisms, but not in earlier ones, and are called derived characters. So as you go through a cladogram through time, you're seeing more and more of these derived characters. The more derived characters a species has, the further along its place is on the cladogram. The cladogram indicates an approximate order of evolution. So evolution A or E, in this case on the diagram below, evolution of species E happened first and then the others happened later. A phylogenetic tree, that was the word I couldn't think of, not only indicates the order of evolution, but also indicates sort of how much time has passed. So on a phylogenetic tree, the way it's supposed to be is that it's supposed to have really long lines if there's been a super long amount of time that has passed, and very short lines if only a tiny amount of time has passed. Down below, you see 3 different shapes of the cladogram, and they're all identical. So cladograms can be drawn this way, this way, this way. They can be drawn with angles, how am I trying to say that? With right angles, or they can be drawn at not right angles. Okay, I'm having trouble talking. Other things about cladograms, outgroups, you need to know the word outgroup. That's a word that's on your learning targets. Outgroups are the least related to other groups. So take a look at the cladogram down here. I've labeled the outgroup. A and B are more closely related. C and D are more closely related, but all 4 are more closely related to each other than they are to outgroup E. Notice that the trees can rotate around a branch and yet still indicate the same relationships. So take a look at this cladogram. Get my picture out of the way here. Notice the cladogram on the left and the cladogram on the right. The Z, Y, and X have flip-flopped, but it's still showing exactly the same relationship. So you can rotate and it doesn't have anything else, doesn't affect anything. Cladograms, and we call those nodes. Sorry, I just want to point that out. The point where they branch, this right here is the node, and that's what can rotate. Cladograms represent hypotheses, and they are constantly being changed as we get more fossil or DNA information. We update our cladograms. And then this is an opportunity here to show you how to make a cladogram. It's ridiculously simple. This is like a 3rd grade level cladogram, but still makes a point. So you go through and you look at the different animals and you identify what characteristics they have. So for example, slugs have cells. So you would say yes for cells. They don't have hair. They don't have legs. They don't have a backbone. And get my picture out of the way. They definitely do not have opposable thumbs. That would be funny. So you're going to say yes and then no, 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 no. So that means that the slug is going to be super early on our cladogram because it doesn't have these derived characters. As opposed to the human, it has yes for cells, yes for hair, yes for legs, yes for a backbone, and yes for opposable thumbs. So you would put the humans all the way at the end of the cladogram because they're the ones that have all of the derived characters. And then each of the derived characters comes down along the path here, along this main line. that's where you write the derived characters. I'm not going to go through this whole cladogram right now, and you should have done activities like this on the PBS website, so it should be familiar. All right, adaptive radiation. Adaptive radiation occurs when members of the same species migrate into different habitats. So the deer family is a classic example of this. They've moved into all kinds of different habitats. They move up north, they move west, they move south, and different traits are favorable in different habitats. So that gives them an advantage. And remember that an advantageous mutation is called an adaptation. So those with the best adaptations in a particular habitat, they reproduce more and their particular traits become more common. The extinction of one species, related or not related, doesn't matter, may open up a niche for another species to take over. And so that allows more adaptive radiation. And then just a quick FYI, the term divergent evolution, I was trying to look online to see what the real difference is between adaptive radiation and divergent evolution. Both of them indicate the idea of having a common ancestor and splitting into new species from that common ancestor. Do you need to know the difference between the two terms? And I think the answer is no. If you read a problem that says something about adaptive radiation, or you read a problem and it says something about divergent evolution, it's pretty much going to be the same problem. It's not going to be anything different. So I'm going to tell you, for now, let's just consider them synonyms. So the evolution of two or more species from a common ancestor is divergent evolution. All right, so how do new species form? So we've already talked about natural selection. We're kind of giving you the idea. This is another way of saying it a little bit more detailed. A local population gets separated from others of its species. So what we see in the picture below is an experiment, actually, where they were purposely separated from one another, and they were all the same. They were all the same species initially. Different mutations or adaptations are beneficial in this new environment. So as they go into new environments, maybe they have characteristics, some have characteristics that allow them to reproduce more. They eat better, they're healthier, they escape predators more often, whatever, they reproduce more. Over time, they become what are called ecological races. So they can still mate, but they're separated and they tend to prefer individuals like themselves, but they're not officially a new species yet. So differences in morphology and DNA start to become significant at this point. Eventually, hybrids, and a hybrid is when two of these ecological races, when they mate, they're not as successful as the separated. And this is part of why the organisms prefer to mate with kinds that are like themselves. And finally, the two populations become incapable of interbreeding successfully. And that might mean that they can't physically mate, or that might mean that if they do mate, their offspring don't survive well. So let's talk about the concept of reproductive isolation. Reproductive isolation, this is part of what leads to speciation, are barriers that reduce the gene flow between different species by preventing them from producing offspring or ensuring that hybrid offspring are sterile. Just a couple quick definitions. Remember, hybrid is a cross between two species. A zygote is a fertilized egg. So when sperm and egg come together, that's a zygote. Pre-zygotic isolating mechanisms, that's what we're talking about on the top here. That means mechanisms that prevent the organisms from ever mating. So for example, geological distance. If they're not anywhere near each other, lions and tigers, for example, lions live in Africa, tigers live in Asia. They never come in contact with each other except in zoos. So they don't mate with each other, but they are capable of mating with each other. But that would be pre-zygotic because the zygote never forms. And then post-zygotic mechanisms are when organisms are capable of mating with one another, but their hybrids don't survive as well. All right, so, and actually, let me show you some of the examples here, because these are totally things you would need to know. So pre-zygotic would include temporal, ecological, behavioral, mechanical mechanisms, and there's more than this. So temporal implies that they are not fertile at the same time of year. So in this case, these frogs like to mate in the daytime, in the summertime, and these frogs they mate later in the year. You might see flowers that come into bloom in the early spring, some bloom in the middle of the summer, some bloom in the late fall. That's temporal isolation. That's their pollen that's being made, and they can't mate with each other if they're fertile at different times of year. Ecological just means that they're in different areas. Sometimes that might mean, so the lion and the tiger, they're on different continents, but it might also refer to some species live in the grass and some species live in the trees, and so they don't mate with one another. Behavioral is one of my most fascinating ones. Behavioral is especially common in birds, where two species live right near each other, eat the same foods, nest in the same ways, but if they don't sing the right song, they never mate with each other. So their songs are very specific, or they have special mating dances. And if they don't dance the right dance, they don't get mated with. So that's what separates these species. And then mechanical refers to just not being able to physically fit together in order to mate. So the example here is the Great Dane and the Chihuahua. They are mechanically isolated from one another. Hybrid, so post-zygotic isolating mechanisms, that's at the bottom. That refers to the hybrids not doing well. So inviability means that they don't develop to reproductive maturity, so they either die as a zygote, or when they are born with mutations or whatever. They just don't survive well. Infertility means that they do survive, but they are not able to have offspring of their own. So the classic example of that is the mule, which is a crossbreed between horses and donkeys. They develop perfectly, but they're sterile. They can't have offspring of their own. And then hybrid breakdown. The F1 hybrids are fertile, but eventually they're not fertile anymore. Their offspring are not fertile, and so eventually that species can never be a species of its own. So the idea of a species is that it can reproduce all by itself and nobody else can reproduce with it. All right, Sympatric and Allopatric speciation. These are terms that are directly on your learning targets. In allopatric speciation, take a look at the diagram. There is a geographical barrier that leads to reproductive isolation. So the lions and tigers have experienced allopatric speciation. Each population experiences separate selective pressures. The dominant form of speciation in organisms that use sexual reproduction is allopatric. They have separated by habitat, and they no longer mate because they're not near each other anymore. And these overlap with the reproductive isolation, so you might notice some similarities between the two. Okay, and then sympatric speciation is the evolution of a new species from an ancestral species while remaining in the same geographic location. So notice the trees that are forming there, and that they're forming in the same location, but you have two varieties of trees. All right, convergent evolution. This one's weird. This one is exactly kind of the opposite of divergent evolution and adaptive radiation. This is when you have species that are not closely related, but they end up looking morphologically, their shape is similar to one another. And that's because they live in similar environments in different areas. Someone's at my door again, so give me just a second here. Okay, keep getting interrupted here. So anyway, they tend to look like each other over time because similar mutations are beneficial in these similar environments. The classic example is the shark and the dolphin. Sharks are fish. They look like they do because they've always lived in the ocean. Dolphins are mammals. They believe that they were four-legged organisms that moved into the ocean over time with mutations. And streamlined mutations, losing the back legs, for example, was advantageous for it. And so it looks like a shark, but when you dissect them, like we mentioned earlier, if you dissect the flipper of a dolphin, you find a humerus, a radius, an ulna, and phalanges, and you don't find that in the shark. So that's an example of two species that look a lot alike, but they are not closely related, and we call that convergent evolution in that circumstance. All right, and for some reason, this is not moving forward. There we go. All right, and then the last thing that I love these bells. The last thing that I wanted to mention was sexual selection. This occurs when members of one gender, it's usually the females, choose mates based on a specific trait or behavior. Females have a limited number of eggs or offspring, and so males can have millions of sperm in any given sample, whereas females only have 1 to a dozen eggs. So females tend to be pickier about whom they're going to mate with because they've invested a lot of energy and to raise the offspring, even if they're not raising them specifically, just within their bodies to grow them. It takes so much energy to feed these babies. That they're very particular with who they're going to mate with to increase the odds of their offspring survival. So the trait that females are searching for, it should be a trait that is an indicator of the overall health or fitness of the male. You should definitely watch these videos because they are fascinating. But one that I'll mention specifically, the tail of the peacock. Having a peacock's tail is a huge disadvantage. The males don't fly well. They're super obvious to predators. The females are absolutely looking at the tails, and they're choosing the males with the biggest tails. In fact, they even have discovered that the females identify how many spots are in the male's tail, and they won't mate with one that doesn't have enough spots. So the question is, why on earth would these females choose them? Well, the idea here that Darwin came up with is that the male who has this outrageous tail, he must be really healthy. He must be eating well because he's able to grow this tail. He must not be sickly because he's able to grow this tail. And he must be able to escape from predators if he was able to grow this tail. So he's actually a very fit sample, I guess. And so the females are interested in mating with those males. All right, ladies and gentlemen, that is it. Let me know if you have any questions.